The overall goal of the following experiment is to use various virological and biochemical approaches to dissect the role of a host signaling pathway in the lytic replication of a model herpesvirus. Here, as an example, we examine the role of the mitochondrial antiviral signaling protein, MAVS, during infection with herpesvirus gamma HV68. First, to examine the role of MAVS during acute infection, wild-type and MAVS knockout mice are infected with gamma HV68. Following an acute viral infection, the mouse lung is collected, and viral loads are determined by a plaque assay in which the samples are serially diluted and plated on monolayers. The number of plaques is then counted. Increased viral loads in the knockout mice suggest that the gene of interest plays an antiviral role. By contrast, reduced viral loads in knockout mice indicate that the gene is required for viral infection. To validate these findings in vitro, viral multi-step growth kinetics are examined in mouse embryonic fibroblasts. Wild-type and MAVS knockout mouse embryonic fibroblasts are infected with gamma HV68 and incubated for various times. The cells are then collected and plaque assays are performed to compare multi-step growth kinetics between wild-type cells and MAVS deficient cells. To further validate these findings, wild-type and MAVS knockout mouse embryonic fibroblast cells are infected with gamma HV68. Then, DNA and RNA are separately extracted from the infected cells, and the viral genomic transcripts are analyzed by real-time PCR.
We first had the idea for this protocol when we studied the role of a host innate immunity pathway during a gamma herpesvirus lytic infection. This protocol can be potentially applied to investigate the roles of host signal.